All right, we can go down here, upstream regulators. Let's go to, I always like to start out with disease and functions. I think this is a kind of a good background for all this stuff. So click on this tab. I hate when they size it by the significance of the finding. So these are biological functions. You can see here, neoplasia, epithelial tissues, tumor genesis. What this is telling me in the language is this is a heck of a lot of cell growth going on. I like to size it by number of genes. So why don't you do that? And you can see a lot of gastrointestinal. What are they trying to build eventually with these cells? Does anybody remember? Endoderm, is that what you said? Even if you didn't, you just pretend you did. All right, cool. Good job. You're extra credit. Awesome. Yes. They're trying to build an endoderm, you know, the inside of the stomach. Look at all this stuff. You can see a lot of gastro stuff. A lot of digestive system. Some of this stuff goes down, which is interesting. So let's see. Let's take one that's a lot of transcription. Again, gastrointestinal. Here's what it's interesting. So remember, what's our fold change that we're looking at here? What are we comparing? What is this fold change based on? Say that again. Okay, day and cell type is the factors that we want to find. But we put in the fold changes based on iPS cells, the pluripotent, versus the stem cells. So everything here is reflected on the pluripotent state. Here's what I want you to notice, is if you look at cell death, there's a lot more cell death in the pluripotent cells than the stem cells, embryonic. That's a big deal if you work in stem cells. Stem cells are supposed to last forever, right? You're supposed to use it to regrow everything, you know, like the Energizer bunnies of the human body. What we are finding, and as I've kind of looked through this data, is I've noticed that the cells from the adult are much older and therefore have a shorter shelf life. Not only that, but they have some kind of cancerous properties as well. What you'll find is, you know, as I look through here, I'm not sure if I want to put these things in my body, right? Put a pluripotent in and then you get a tumor. That wouldn't be too cool. Also here you'll notice, go to these blue things. Cellular assembly, organization of cytoplasm. These sections are blue, which would mean that these are predicted to be going down. So the cytoskeleton is important in cell movement, it's important in cell adhesion. And what you're going to find as we kind of go through these lists, you're going to find a lot of cell adhesion, cell movement stuff. So again, this is, I'm going to click on one of these. Organization aside, no, let's see, which one? Which one has the biggest Z score? Ooh, Microtubule Dynamics. I'm going to click on this box. There it is. As I keep going down and down, what happens is I kind of go into that area, and basically what this is doing is it's producing all those genes in our gene list, those 1,000 genes that we put in that are associated with microtubule dynamics. Not only that, but it tells me here are all the genes associated with this particular biological function based on its fold change. So this is in the pluripotent cells. Based on the literature, this is known to decrease this process. And you can see as you go down, most of these are definitely decreased. The number of evidence showing that this process is probably decreases versus increased is outnumbered to the decreased part. Therefore, the software is saying, giving it a confidence score, saying, based on these results and what we know about this biological system and these genes, we predict this function to be more inhibited. The more negative this z-score is, the more certain the software is that that is either inhibited or activated. Does everybody understand that? You can think of these z-scores as almost like standard deviation, right? Random noise would be 0. Evidence that points to more activation is going to be more positive. The software says if the evidence for activation is more than 2 standard deviations above random noise, it's going to say, yeah, that thing is activated. If it's below -2 standard deviations from just random noise, it's going to say it's inhibited. Does everybody understand that? Cool. Either I'm a great teacher or you're just lying to me. It's all right. Go ahead, lie to me. I don't care. I've had a hard week. If you wanted to find out the evidence to say, okay, GAB1 is predicted to decrease this process when it goes down, I could click on this 6. That means there's 6 pieces of evidence in their database to show that GAB1 protein increases or basically decreases microtubule organization. And I can click on any of these papers. That is a cool part of this. And that's one thing you can use with IPA, not just to analyze gene sets, but to actually do some investigative stuff yourself, right? If you have a gene you're interested in, you can pop it in here. What pathways are it in? What does this software know about this? This is a great resource. This is like Google for genes, in a way. Except better. It's got Java. Okay. You can get out of that. Over here, you'll see this up one level. Just click in that until you get to the original page. Okay, let's go down here. So I took my cursor and I brought this window up a bit. Here are all the biological functions that are overrepresented in our gene list. Remember, we're pulling out more than we would expect by random chance of genes associated with these biological functions in our 1,000 genes. They are ranked by p-values. So again, this is the probability that I would get all of these genes by random chance. Here is the activation score if one can be given. And this is basically saying, okay, we know that this pathway is overrepresented. I mean, the odds of getting this is 3 times 10 to the negative 16th, these genes. But based on the expression of these genes, whether they go up or down, we'll make an activation score. And we can sort either by p-value. And here's the thing is, look at these pathways, right? We're talking about the endoderm. This is the abdominal region. You've got, stem cells. Stem cells proliferate. That's what they're good at. Number one thing is proliferation of cells. Next one, cell movement, migration of cells. Abdominal neoplasia. Neoplasia is the basically formation of skin. Invasion of cells. Yeah, apoptosis down here, cell survival. This is like your hot list, this is like your mixtape of, pluripotent versus stem cells, or embryonic stem cells. We can also sort by activation state. Let's do that. Now this is telling you, we've got a list, we know that all these functions are associated. Now we're getting an idea of what these functions are doing in our system. How are they different between pluripotent versus embryonic? Well, I would say pluripotent stem cells have decreased cell-to-cell adhesion. And that's important. This means that these are less sticky than embryonic stem cells. What you'll know, or if you so I actually did some reading. I'm learning a lot about stem cells, actually. I know a little bit about just about everything at this point. I've worked at so many studies that you can ask me any disease. I'm like, oh yeah, I remember that. NF kappa B, that's what it is. So we probably have less cell adhesion. What this is telling me is that these pluripotent cells are less sticky, which is kind of more of a differentiation state. Does that make sense? Embryonic stem cells are very, very sticky. The blastulas, you know what I'm talking about? They make these formations, these, groups of cells. They have to be very sticky. They have to stick to each other. Okay, let's hit this activation state again. What this is telling me up here is there's a lot of transcription going on in these pluripotent cells. That also tells me that these pluripotent stem cells are probably more differentiated than our embryonic stem cells. That embryonic stem cells probably don't produce a lot of genes, or probably produce less genes, or have a product from less genes, is because they're undifferentiated. They don't have to specialize. They don't have to turn on these genes. What this is showing me is that the pluripotent stem cells are probably producing more genes than our embryonic. Therefore, they're probably more differentiated. Also, what was the other one? That these, even though they are less sticky, they probably are moving more. Cell movement of endothelial cells. Again, this is basically kind of a flavoring our gene list. This is giving me the background on our criminals, basically. Now I'm going after the gangs next. Then I'm going for the big guys, the godfathers after that. Okay, so stay with me. Okay, disease and functions. We got everybody's criminal history, basically. That was interesting. I kind of understand what's going on. Let's go to canonical pathways.